Hello, so I'd like to solve a sample two-point mapping problem for you. A situation where we're dealing with a pair of genes, but in this case we need to be open to the possibility that the gene pair may be on the same chromosome and not segregating independently. So probably work best for me to just do a sample problem. So here's the problem. So let us imagine we are dealing with a gene pair in tomato, and let us say that red fruit color is controlled by the dominant allele of the gene labeled T, and tangerine fruit color is controlled by two recessive alleles of the gene T. And at another locus, and in this case the problem tells me that the other locus is on the same chromosome. The other locus, a gene big H, the dominant allele, is responsible for smooth leaf phenotype, and the recessive allele is responsible for the hairy leaf phenotype. And the question tells me that both of these genes are on chromosome 7, and they are 20 map units apart. So imagine that in this problem, a breeder takes a pure breeding tangerine colored tangerine and smooth-leaved tomato plant, and crosses it with a red fruit colored plant, a pure breeding plant, excuse me, so that's homozygous dominant, and crosses it with a red fruited tomato that had hairy leaves. And the F1 plants, which were double heterozygotes, and we would expect those to be red fruited and smooth leaved. And they were test crossed with a homozygous recessive tangerine fruit colored plant that had hairy leaves. And so I'm going to write the test cross alleles in a different color because we kind of can set those aside. The function of the test cross is to allow us to simply observe the segregation of the alleles in the heterozygous plant that were of interest. So the thing we need to think about when we are considering the progeny of this cross, imagine we're asked the genotypes and phenotypes in the progeny of this cross. We need to have a think about what different genotypes and phenotypes would we expect. So it's a test cross, so the genotypic and phenotypic ratio will be identical. We would expect 4 different types of progeny in this cross. We would expect there to be progeny that are red fruited and smooth leaved. We would expect there to be, I think this is a little t. We would expect there to be tangerine fruit colored and hairy leaves. We would expect tangerine fruit color and smooth leaves, and we would expect there to be red fruit color and hairy leaves. And if it were the case, I'm just going to clean this one up to make sure there's no confusion, so I'm just going to make a bit of a mess here. So bear with me. Just to make sure there's no confusion. So these are the 4 possible genotypes we would expect from a test cross of a dihybrid. Now, if these genes were independently assorting, we would expect an equal proportion of all 4 of these types of progeny. However, we are told that they are linked 20 map units apart. So we would expect there to be 80% of the offspring to have the parental type. Now, we can't always assume that the parental type will have both the dominant alleles and both of the recessive alleles. We need to have a think about the phase of linkage. So I'm going to illustrate that for you, and we're going to have a little consideration of what's going on in this plant here. So I'm going to draw you a picture. So this plant here arose from a chromosome that was contributed from this parent, which had the recessive allele for fruit color linked to the dominant allele for hairy or smooth leaves. And the second chromosomal contribution came from this parent, which had the dominant allele controlling fruit color linked to the recessive allele for smoother hairy leaves. So this here is what the meiosis looked like in this F1 dihybrid here. The phase of linkage is repulsion. The genes are coupled in repulsion. We would expect, therefore, that the majority of the offspring, the majority of the gametes produced from this parent would have one of these combinations. Indeed, we would expect 80% to have one of these combinations. So we would expect 80% to look like these progeny here, and we would expect 20% to be the product of recombination, because the definition of 20 map units between these two loci means that 20% of the products of this meiosis will result from a crossover, which will shift the two dominant alleles to be inherited together, here they are here, and the two recessive alleles to be inherited together. So the answer to this problem is we would expect 10% of the progeny to have this genotype and both dominant phenotypes. The other 10% would have this genotype, both recessive phenotypes. We would expect 40% of the progeny to have this tangerine fruit color and smooth leaf, and the other 40% to have this red fruit color and hairy leaf. So that's how we would tackle this problem.